Good afternoon. It's a pleasure to be able to replace Svenja today. Now, you've heard a lot about macroanatomical landmarks, and the question is, how do they relate to microscopically identifiable borders between cortical areas? And as with most things in life in anatomy, you've seldom got a black and white situation. So if you ask the question, is there a correlation between the position of macroanatomical landmarks and that of borders between cortical borders? The answers are yes and no. Depending on the part of the area of the brain that you're in, depending on the area that you're examining, there is a correlation, maybe, or maybe there isn't. This is something that we can't state categorically a priori. Now, examples of macroanatomical landmarks that provide a good hint as to the position of microanatomical borders are provided in this slide here. So on the left side of the image, you've got the mid-fusiform gyrus, which is highlighted by the white lines on this reconstruction of a human brain, and this is a view on the occipital lobe, the bottom part of the occipital lobe. And color coding is for cytoarchitectonically identified areas, FG1 in green and FG2 in the violet color. The panel above shows a cytoarchitectonical section, a coronal section through this fusiform gyrus with areas FG1 and FG2. And the borders of FG1 and FG2 on this cytoarchitectonical section are highlighted by the arrowheads. And the border between FG1 and FG2 is located within the mid-fusiform gyrus. Other studies have examined, analyzed the correlation between macroanatomical borders and macroanatomical landmarks and microscopical borders to see how often you can find a correlation. And the box plot shows that when you're analyzing primary visual cortex, primary motor cortex, there's actually a relatively good correlation between the macroanatomical landmark and the position of the border. So the landmark would provide a good hint as to the position of the border. But when you move up to hierarchically higher areas, such as areas 44 or 45 of the Broca region, this correlation between macroanatomy and microanatomy becomes less tight. The bottom part of the slide shows the correlation between the borders of the primary visual cortex as identified in cytoarchitectonical studies and a functional imaging study. And the data from the cytoarchitecture and from the functional imaging study were brought into the same reference space, and the distance of the borders to the central to the calcarine sulcus, in which the primary visual cortex is localized, was measured. And you can see that when you're in the calcarine sulcus, most of the voxels are primary visual cortex. As you move out of the calcarine sulcus, you still find some voxels which can be identified as primary visual cortex. So this leads then to the next point, the cases in which macroanatomical landmarks are not helpful to identify borders, microanatomical borders between areas. So again, two examples. The left side would be the Broca region, areas 44 and 45. So you here see reconstructions of 4 different hemispheres. The top row are left hemisphere from 2 different individuals, and the bottom row of images are the right hemisphere from 2 different individuals. Red codes for area 44, as you can see it on the surface of the hemisphere, and yellow codes for area 45. And this pink arrow highlights the position of the diagonal sulcus. And you can see that in this top left example, the diagonal sulcus is located within Brodmann's area 44. In this example here, the diagonal sulcus is very helpful because it marks the position of the border between areas 44 and 45, as it does in this example here. But in this fourth hemisphere, variability is such that it actually does not have a diagonal sulcus. So if you're looking for your macroanatomical landmark to identify the microscopical border, you're lost here. Again, cases in which macroscopical landmarks just don't exist would be this example here, showing the subgenual cortex, so medial surface of the hemisphere below the corpus callosum, and here you have absolutely no sulcus, which identifies the borders between Brodmann's areas 33 or subdivisions of his area 25P and the orbitofrontal cortex. Now, the next couple of slides, I'd like to walk you through a couple of further examples of how we can find a correlation or a lack thereof between macroanatomy and microanatomy. And I'd like to do it with some examples of borders that are actually quite easy to see, such as the border between the primary visual cortex V1 and the adjacent secondary visual cortex, and then borders that are not always that easy to see, such as those between areas 3B, for example, and area 1 of the somatosensory cortex. So starting with the primary visual cortex, this slide shows a coronal section through a human occipital lobe processed for the visualization of cell bodies. I've labeled some sulci that Julian already mentioned. So there's the intraparietal sulcus. We've got the parieto-occipital sulcus, which can be sectioned multiple times in a coronal section. There is the calcarine sulcus down here, and the lingual sulcus. And then I've labeled a couple of sulci with interrogation signs, because we actually, if we look at this coronal section here, and even if we follow it through a couple of sections, depending on how many you've got available, you don't know if this is a side branch, for example, of the parieto-occipital sulcus, or if it's maybe a sulcus that's only present in a low percentage of brains that have been analyzed. If we now zoom in to this area highlighted with the red box, which is the position at which in this section the border between V1 and V2 is at a higher magnification, you can see here on the surface of the brain, the medial surface, we've got V1, and going into the lingual sulcus, we've got V2. If we go into an even higher magnification, you can see in V1 the subdivisions of layer 4, which are characteristic for this area. It's the only brain area in which layer 4 can be subdivided. Within the lingual sulcus, we've got V2 with extremely large neurons identifiable in the deep part of layer 3, so in layer 3C, these are the so-called Maynard cells. These differences in cytoarchitecture between V1 and V2 make it very easy to identify the position of the border between these two areas. This second example is again a coronal section processed for cell body staining through a human hemisphere, this time at the level of the central sulcus, which is labeled in the slide with CS. You can see the precentral gyrus and the postcentral gyrus. depending on how your section has been taken, the hint that the precentral gyrus is thicker than the postcentral gyrus is unfortunately not always helpful. What is helpful is the fact that cortex on the precentral gyrus is a lot thicker than that on the precentral gyrus. So this is one of the cases where when you move into individual anatomy, apart from the individual variability in the presence or absence of certain sulci, these general aspects of macroanatomy aren't always helpful. If we zoom into the central sulcus, we can see here on the precentral gyrus cortex from area 4, the primary motor cortex. It's followed by area 3A and 3B. If we zoom in even more to the motor cortex, we can see enormous cells in layer 5 of area 4. These are the bed cells. These are the largest cells in the human brain, and their neuronal bodies can reach up to 100 micrometers in diameter. If we now move within the central sulcus, but over to its other bank, we've got area 3B and area 1, and here the border between both areas is located within the central sulcus. So again, we don't have a macroanatomical landmark to identify reliably the position of this border. So summarizing up to now, we have a microanatomical segregation within the brain. We've got areas with very, very different laminar distribution patterns of their neuronal cell bodies, and we don't always have reliable macroanatomical landmarks to highlight the position of borders between these areas. So what we need is a map of microscopical landmarks, and you can say, well, such a map already exists. Brodmann's map has been around since 1909, and it's been very helpful to identify or to assign labels to functional imaging activations. The problem with Brodmann's map is that it's based on visual inspection of just one hemisphere, so it doesn't tell us anything about inter-individual variability. We nowadays also know that he underestimated the number of areas that the cortex can be parcellated into. Unfortunately, also it contains areas that really don't exist. Because, for example, Brodmann's area 19, which is this part of the cortex here on the medial, on the lateral surface, and this part here on the medial surface, it doesn't exist in the sense that even if you add up all the functional imaging studies or all the different microstructural studies carried out in this part of the brain, and you put them together like a puzzle, you're not going to obtain Brodmann's area 19. So obviously here he made some sort of a mistake, basically. And the other problem is that Brodmann's map is a two-dimensional schematic drawing, and the brain is three-dimensional. Now, you might argue that Talarajan-Tourneau brought Brodmann's map into 3D, so that problem's also been solved. But if you look at their drawings carefully, you can see that, okay, they identified Brodmann's areas on the tips of the gyri, but they actually did not show the position of the borders between these areas. So that's why we need to create a new map of the brain. And when you come to identifying microscopical borders, this example, again, the one between area 17, V1, and area 18 of Brodmann, V2, you can see it's very easy to identify the border. But if we move into hierarchically higher areas, the differences become so subtle that you start having problems. So if I didn't have this arrowhead here, highlighting the position of the border between areas 46 and FP1, which is part of Brodmann's frontopolar area 10, you probably wouldn't be able to see it. So that's why modern microanatomical studies aiming at parcellating the brain have decided to take an approach which would avoid observer-dependent definition of borders. And that's important because depending on the observer, you can identify a different number of areas within the same part of the brain, just depending on the criteria that you use to identify a border. And this is highlighted by this slide here, in which the map on the top left is that of Brodmann, and in green is his anterior cingulate region, which contains 4 areas. Areas 25, 33, 24, and 32. But at the position at which Brodmann only identified 4 areas, other anatomists have been able to identify up to 24 areas. So that's why modern approaches have tried to quantify cytoarchitectonic criteria. And the most important criterion which is used in cytoarchitectonic brain mapping is the laminar distribution of the cell bodies within a cortical area and the packing density of these cell bodies in the different layers. We also look at the relative thicknesses of each of the layers compared to the total cortical thickness, or the presence of clearly identifiable sublayers between these layers. But basically, the laminar distribution packing density is the prevailing criterion. And this can be quantified by the gray level index, in which histological sections are scanned, and you set a threshold with which you can separate the cell bodies from the neuropil. So you create a binary image, and if you set a grid over this binary image and then calculate the volume fraction of cell bodies compared to total brain volume within this sector of the grid, you get the gray level index value for that position in the grid. And this gray level index value can vary from 0%, so you've got no cell body at that position, to 100%, which would be that this part of the cortex is completely occupied by cell bodies. The second aspect of this observer-independent mapping is that we can also test the position of borders that we've identified for statistical significance. The image shows a coronal section through the subgenual portion of the cingulate gyrus, again, a cell body staining. We can transform this image here into a gray level index, and if we then draw contour lines separating layers 1 and 2 and then layer 6 from the white matter, and we extract GLI profiles perpendicularly to these lines, we can measure the changes in the gray level index along each one of these profiles. And if we now compare the profiles from position A and position B, you can see that they're very different in shape. And we can quantify the differences in these profiles. We use the Mahalanobis distance to quantify how different these two profiles are. So if a large Mahalanobis profile, Mahalanobis distance would mean that the two profiles that you're comparing are more different than if you've got a small Mahalanobis distance. Now, we don't just compare pairs of profiles arbitrarily taken from the cortex. What we do is we systematically compare pairs of profiles or adjacent blocks of profiles along the cortical ribbon, and for each one of these comparisons we calculate a Mahalanobis distance, and that's how we obtain a Mahalanobis plot. And then you see that there are different peaks along this plot here. The peaks would indicate putative borders, and if we then test these peaks for significance, that is then the indication of a cortical border. And for this specific example here, what you can see is that we've identified Brodmann's area 33, we've identified his area 32, and within his area 24, at this position here, we've been able to identify a subdivision into an S24A and an S24B. So we decided to reassess Brodmann's anterior cingulate region. This slide here shows a series of coronal sections through the left hemisphere of a specific brain, a post-mortem human brain. And we were able to identify a total of 21 different areas within this region, which Brodmann only identified 4 areas. So we've got a new map of the anterior cingulate cortex with these 21 areas, and we can also identify a total of 3 regions: the subgenual anterior cingulate cortex, pregenual anterior cingulate cortex, and then anterior and posterior subdivisions of mid-cingulate cortex. Now, I said the brain isn't just in 2D, it's in 3D, so what we want to do is to bring it into 3D to be able to study variability in the size and the location of the brain relationship with macroanatomical landmarks. If we overlay these maps from different individual brains, we can obtain a maximum probability map, which can be used for overlay with functional imaging studies to identify the microanatomical area that subserves the function of the area that you might be examining in your functional imaging studies, or which can also be used to carry out meta-analysis of functional imaging studies, and then more comprehensively characterize the area which was cytotectonically identified. And with these comments, I'd just like to finish my talk for today. And thank you for your attention.
Any questions to Nicola?
Hi, very nice work, especially with the fact that it follows the cingulate round the bend. I was wondering, do you have any ways of dealing with the fact that your sections, if they're, are they all coronal? And if so, how do you follow the bend round the cingulate?
That mapping study was actually carried out with a total of 10 post-mortem brains, 6 of which were coronally sectioned and 4 horizontally sectioned. So when you're looking for, you can follow all the borders. You have some sections tangentially sectioned around the knee of the corpus callosum when you've got the coronal ones, and you've got, of course, problems with the ones above the corpus callosum when you've got the horizontal ones, but you infer from both.
Got 2 quick questions. The first is that are there any areas in the cortex that are in different lobes, but actually they look cytoarchitectonically identical? So if you give a histologist 2 areas blind, would you always be able to pinpoint that is in the frontal lobe, right, or actually cytoarchitectonically there are areas that actually look identical?
You have trends within the lobe. So for example, if you're in the cingulate lobe, you've got a very, very prominent layer 5, and not such a nice layer 3. If you move into the parietal lobe, you have very large cells in deeper layer 3, and your layer 5 isn't that prominent, so you've got a shift in the ratio and size of cells. You've got a much more prominent layer 3 in the parietal cortex. You also have a very prominent layer 4 though in prefrontal cortex. So as I started my talk with a yes and a no, here it's the same thing. There are some aspects that are typical of a lobe, but others that unfortunately aren't, that are the same over various lobes.
And Nicola, for the sake of being educational, right? Another question, very quick. Brodmann areas is a complete work, but imperfect.
Yes.
The work that you're doing is the cutting edge, but incomplete. So, what is your advice for the young neuroimagers? Should we, when we try to map the cortical area, should we use a kind of hybrid of the two? So when we found your areas, we should use your areas. When we don't, we should really go back to Brodmann. Should we wait for you to complete? So what is the advice?
I'd say exactly what you said. I'd say that if cytoarchitectonically identified areas, or not cytoarchitectonically identified areas, areas with cortical borders identified with an observer-independent method, if that is available for the part of the brain that you're examining, so if you have probability maps, and these are freely available via the U-Brain Atlas and via the anatomy toolbox for SPM analysis or for free surfer analysis. So if you've got the anatomy toolbox, use it. As you say, the work is imperfect in the sense that it's not complete yet. So at least, but you know, even in the anatomy toolbox, if there's not a cytoarchitectonically identified area at the location at which we are, the toolbox does tell you the gyrus in which you are. So then at least provide that gyrus, because it is important to pinpoint the anatomical structure underlying your activation. And it's important later on because, you know, if your blob is here, it's not the same as it being up here. You know, this is cingulate cortex, and this here would be pre-SMA. And if you've got a blob up here and you say, well, this is a cingulate cortical activation, you're actually giving the future scientists a very wrong impression when they look for functions in which cingulate cortex is involved, for example.
By the way, more than 2/3 of the cortical surface are already mapped with the observer-independent method, so that 1/3 is not mapped up to now, but the restrictions by the 2/3 is so big that the assignment to gyrus, sulcus, or a Brodmann area is more and more definite. Yeah. Could you come to a micro? Because this is recorded, so you can see the whole course later on the website, and you should also have a chance to hear questions and answers.
In the parcellation article by Glasser and Van Essen last June, they look at multimodal approach to dividing up the cortex, including myelination, cytoarchitectonics, etc. And one of the things that they caution is that the sulci and gyri do not correlate consistently with the cytoarchitectonics or the parcellation that they did. So how do you quantify the probabilistic odds that the cytoarchitecture that you're talking about will be in that gyrus?
probability maps are in 3 dimensions, so we can look at the 3D volumes of our cytoarchitectonic areas and assign them to a certain sulcus or gyrus. In addition to that, you mentioned multimodality. Today I only spoke about cytoarchitecture, but actually the borders of these 21 subdivisions within Rothman's anterior cingulate cortex are also revealed by differences in receptor architecture. So this map here isn't just a unimodal map, it's also a result of multimodal analysis. Plus, we have the cytoarchitecture in 3D and can then correlate it 100% with macroanatomical landmarks.